Having identified the genetic material as the nucleic acid DNA or RNA, we proceed to examine the chemical structure of these molecules. Generally, the nucleic acids are a long chain or polymer of repeating subunits called nucleotides. Each nucleotide subunit is composed of three parts: nitrogenous base, 5-carbon sugar, pentose, and a phosphate group. There are pentose sugar. There are two types of nucleic acids depending on the type of pentose sugar. Those containing deoxyribonucleic sugar are called deoxyribonucleic acid, and those with ribose sugar are not ribonucleic acid. DNA is found in the nucleus of eukaryotes and nucleus of prokaryotes. The only difference between those two sugars is that there is one oxygen atom less in deoxyribose. Nitrogenous bases. The bases are nitrogen-containing molecules having the chemical properties of a base, a substance that accepts H plus ion or proton in solution. DNA and RNA have both four bases, two purines and two pyrimidines in their nucleotide chain. Two of the bases, adenine and guanine, have double carbon nitrogen ring structures and are called purines. The bases thymine, the cytosine C and uracil U have a single ring structure, and these are called pyrimidines. Thymine is unique for DNA, while uracil is unique for RNA. The phosphate functional group, it is derived from phosphoric acid H3PO4, has three active OH- groups, of which two are involved in strand formation. The phosphate functional group PO4 gives DNA and RNA the property of an acid, a substance that releases an H plus ion or proton in solution at physiological pH, hence the name nucleic acid. The bonds that are formed from phosphates are esters. The oxygen atom of the phosphate group is negatively charged after the formation of phosphodiester bonds. This negatively charged phosphate ensures the retention of nucleic acid within the cell or nuclear membrane. Nucleoside and nucleotide. The nitrogenous base is chemically linked to one molecule of sugar at the one carbon of the sugar, forming a nucleoside. When a phosphate group is attached to the 5' carbon of this 5' carbon of the same sugar, the nucleoside becomes a nucleotide. The nucleotides are joined by condensation reaction to form a polynucleotide chain. The hydroxyl group on the 3' carbon of a sugar of one nucleotide forms an ester with the phosphate of another nucleotide. The chemical bonds that link the sugar components of adjacent nucleotides are called phosphodiester bond, indicating the polarity of the strand. Based on the X-ray diffraction analysis of Maurice Wilkins and Rosalind Franklin, the double helix model for DNA was proposed by James Watson and Francis Crick in 1953. The highlight was the base pairing between the two strands of polynucleotide chain. This proposition was based on the observations of Erwin Chargaff that adenine pairs with thymine with uh, with two hydrogen bonds, and guanine pairs with cytosine with three hydrogen bonds. The ratios between adenine with thymine and guanine with cytosine are constant and equal. The base pairing confers a unique property to the polynucleotide chain. They are said to be complementary to each other. That is, if the sequence of bases in one strand plate is known, then the sequence on the other strand can be predicted. The salient features of DNA. A typical cell contains about 10 times as much RNA as DNA. The high RNA content is mainly due to the variety of roles played by RNA in the cell.